Prokaryotic cells come from the two domains, bacteria and archaea. And bacteria come in many different arrangements, shapes, and sizes. You have to remember that bacteria are so small, even with our compound light microscope, that they don't really have a lot of features that allow us to distinguish them from one another very easily. So, shape and arrangement are pretty important. We've already talked a little bit about sphere shaped bacteria or cocci, rod shaped bacteria or bacilli, or spiral shaped. But in fact, there's a lot more that we can do in order to discuss or describe bacteria, apart from these three shapes, of course, that also includes how they group together. The arrangement of bacteria describes how they tend to divide, and as a result of how they divide, how they tend to form groups. So, if we look over here at cocci, there's a plane of division here, and because these bacteria are sphere shaped, you can slice them any way, and you're going to have potentially different mechanisms of division. So, for example, if you have one bacterium that splits in two, and grows out from one direction, you're going to end up with diplococci here, diplo being a pair, or streptococci, where strepto means a chain. So you can see if they always divide on the same axis, that they're always going to add on to themselves and make a larger group in a single direction. On the other hand, if you've got bacteria that divide in several different planes, for example, you can end up with the tetrad formation. Or if you have ones that divide in several more different ways, you can end up with Sarcina. Or if you go totally random, you can end up with a random cluster of them, which would be Staphylo. So whenever you see Staphylo as a prefix, of course, that means an irregular cluster formation. Now, when we look at bacilli, you'll see automatically there is less in terms of how these bacteria can arrange, primarily because bacilli always extend along their length and pinch off to make a new cell. They never divide sideways. So you're never going to, for example, have them dividing along their long axis, like they'll never be cut this way. They'll always pinch off this way to form more cells at either pole. So you'll end up with diplobacilli if there's a pair, streptobacilli if they grow into a long chain. And for example here, you'll never see staphylobacilli. They just don't exist because of the way that these particular cells happen to divide. You can also have these kind of little halfers here, which are coccobacillus. They're kind of coccoid, they're kind of bacilloid, but not quite either or. They kind of look like little truncated, kind of in-betweenies. So they're difficult to kind of put into one group or the other, so they end up being a little bit of both. On a test, for example, I might ask you, a pair of sphere-shaped cells is called A, and then give you a variety of options. So you should become familiar with this nomenclature, and just being able to describe bacteria is really important. We're going to do more of this stuff in the laboratory as well, but this is a great introduction to some of these concepts. In addition to cocci and bacilli, which will comprise the majority of bacterial species that we'll observe, we also have Vibrio, which are kind of a little bent, rod-shaped bacterium. We have Spirilla, or Spirillum if we're talking about one, that are kind of like a loose S shape, right? You can kind of see that here. They're a little twisted. But you can also have spirochetes, which are coiled a lot more tightly than the Spirilla, for example. In addition to this, there are quite a few more, which we're not going to spend too much time really stressing about, but I just thought that I would mention that you can have club-shaped bacteria. You can also have filamentous ones as well, star-shaped ones, rectangular. Really, the world of microorganisms and their arrangements is potentially endless depending on what's out there. There may be more that we haven't even discovered yet. In terms of size, there's a great deal of diversity. You can have some bacteria as small as 0.05 micrometers and some as large as 600 micrometers, but most bacteria are between 1 and 10, and a lot of the ones that we'll be looking at in the lab as well will fall within this range. Obviously, these are not things that you need to memorize, but this is just a great diagram to help you to compare the size of bacteria, right? So things like E. coli, streptococcus, we've heard about these quite a lot, compared to a red blood cell, which is one of the smaller eukaryotic cells that we look at under the microscope. And even bacteria are considerably larger than a lot of viruses, so you can see that they're even smaller. right? We did talk about how scanning electron microscopy is required in order to view viruses. We cannot see them with compound light microscopy. So, prokaryotic and eukaryotic cells do share some structural characteristics. All cells have a cytoplasm, all cells have a cell membrane that's comprised of phospholipids, and all cells do contain some form of DNA as their genetic material. They also all have ribosomes, but apart from that, there's not really a lot of similarities beyond. So, if you look at the example here of a prokaryotic cell, you can identify all of those things that I just mentioned, but one really distinguishing factor that really separates most bacteria from most eukaryotes is the presence of not only a cell wall, right? Because plant cells obviously have those and so do fungi, but it's what it's made of. And we've already talked about peptidoglycan as being the primary component. There isn't a single eukaryotic cell out there that has peptidoglycan as part of their cellular structure. So it is exclusive to prokaryotic cells. Prokaryotes also have flagella. If you imagine, a lot of them are single celled and they do need to be able to get around in some cases depending on their environment. And flagella are definitely one way that they're able to do that. Fimbriae and pili are also other kinds of hairs, hair-like projections that you can find on the surface of cells that can also help with motility. But generally speaking, the big difference that's going to separate eukaryotes and prokaryotes is the lack of a nucleus in prokaryotic cells. We've already talked about this. We talk about it all the time, but that is truly the defining characteristic that does separate prokaryotes from eukaryotic cells. Bacteria have what's called an envelope, and the envelope is going to encompass everything that is outside of the cytoplasm that divides the cytoplasmic region from the extracellular environment. Typically, you're going to have cell membranes or plasma membranes in all bacteria. Cell walls are going to be present in most, and a slime layer or capsule will be present in some. So again, that slime layer or capsule is a layer made up primarily of polysaccharides, sometimes protein as well, that is secreted by some cells, but not all. So if a bacterium has all three of these, all three of them are their envelope. If it only has cell walls and a plasma membrane, those two things comprise the envelope. So every bacterium has an envelope, but how many layers or components that it has will vary depending on the species. The plasma membrane in bacteria is going to be a very similar picture to what we already know and love about eukaryotic cells. It is semi-permeable, and it only permits the passage of certain types of molecules through. It's going to contain integral and peripheral carrier proteins that allow for things like nutrient absorption or waste excretion, things like metabolic processes, including respiration, in specialized cells, potentially photosynthesis or something that is similar to, and synthesis of structural components. This diagram is kind of reacquainting you with some of the terminology of different proteins and how they're affiliated with the cell membrane. Like, for example, ones that are integral will go through both leaflets of the plasma membrane. Ones that are peripheral will associate with one leaflet or the other. We also have glycolipids or glycoproteins as well that have a sugar component, as well as a variety of others. What kinds of lipids and what kinds of proteins are present in the cell membrane is going to depend on the bacterial species at hand. So there are some types of proteins that are common to most bacteria, but every one is going to be fairly different. So some species, for example, in terms of their lipid content, living in more extreme environments, lower temperatures or higher temperatures, as I mentioned in an earlier talk, will have differences in terms of the saturation of their lipids. For example, ones that are living in lower temperatures are going to have more unsaturated fatty acids in their structures. Unsaturation, you can see double bonding, is going to cause these lipids to kink. And in terms of the biochemistry of this, if you imagine a lot of phospholipids that are very close together, they have barely any space between them, they're going to tend to freeze more easily. But if we introduce space by kinking out these fatty acid tails, what it does is actually pushes those phospholipids further apart, and in turn creates more space between them, which makes them harder to freeze. In colder temperatures, this is obviously beneficial. So, if you were to take a survey of phospholipids from things that live in temperate conditions, very hot or very cold, you're going to see differences in terms of this fatty acid structure, among other things as well. So, this is one of the adaptations that cells can make in order to adapt to their environment directly. Some of them can even change it on the fly. If temperatures change, for example, if they go from one environment to another, they can totally change up what their lipid composition looks like in order to survive in their new environment. Because don't forget, as a single celled organism, a cell is at the mercy of its environment and doesn't have a lot of other ways to mitigate these large fluctuations in environmental conditions. But it can adapt quite easily at the level of its biochemistry. Prokaryotic cells also have a feature that eukaryotic cells have, which is to incorporate very large, rigid molecules into their membranes to help to stabilize them. In eukaryotic membranes, what we have is the integration of cholesterol, which with its many rings is quite rigid and helps to provide membrane stability to keep these things a little bit firmer. Bacteria don't produce sterols, which cholesterol is considered to be a sterol, of course, but they have other molecules that are similarly structured, and these are called hopanoids. They're sterol-like. If you look at their structure, of course, they have a ton of different rings in them as well, and they're also integrated into the cell membrane to promote stability. Now, the cell walls themselves are what provide bacteria with their structure and a degree of protection from the external environment. The cell wall is typically going to be external to at least one plasma membrane and will be unique depending on the type of prokaryote. The uniqueness will come from some small details of its overall general structure, but all peptidoglycan is made up of protein and sugar. Also important to note here that bacterial and archaeal cell walls are extremely different, right? We talked about how archaea do not contain peptidoglycan at all. So that's obviously a big thing. But in terms of even between species of bacteria, you are going to see some variation. The purpose of the cell wall in bacteria is to offer some kind of protection from the environment. Of course, if you're living in an aquatic system, you're not going to want to be bombarded with different factors that may be present there, whether they are just present as pollutants or released by other cells in terms of some kind of biological warfare, because that's the thing. Bacteria will compete with one another for nutrients and resources. They're also important for the maintenance of their shape and structure, and they can contribute to pathogenicity. When we discuss pathogenicity, it simply means how pathogenic or disease-causing bacteria can be inside of a host. So there are components embedded in the cell wall that can be virulent. They can be virulence factors that cause symptoms in people, depending on how our immune response detects them and responds against them. As a little bit of a side note too, when we're developing things like pharmaceuticals or chemotherapeutics, we're typically looking at cellular wall components because they're going to be some of the most external parts that can be easily targeted and attacked with medications. Over here is a diagram. You can see that we have the plasma membrane underneath or more internal to the peptidoglycan layer. Teichoic acid, lipoteichoic acid, and a variety of protein is going to make up the majority of this structure, and we'll see some more detailed diagrams in the next couple of slides. Peptidoglycan is also known as murine. We're going to call it peptidoglycan for the duration of the course, and it is common in gram-positive and gram-negative cell walls. I did mention earlier on that there are certain types of bacteria that do not contain peptidoglycan, but they are few and far between. Most bacteria do contain both, and that also means that most bacteria can be differentiated as either gram-positive or negative. The composition of peptidoglycan is a mesh-like polymer, so many long chains of sugars and amino acids that come together to form a large net-like network that is very strong and difficult to penetrate. The sugars are N-acetylglucosamine and N-acetylmuramic acid, and the amino acids, interestingly, tend to be of the D formation. If you're familiar with stereochemistry at all, you may know that all of our amino acids that we make as humans are L configuration, but in bacteria, they make deconfiguration amino acids, which are different. So our body doesn't actually know what to do with them. They're considered to be foreign. So when our body sees them, it doesn't really know how to process them or break them down well. So it's an added layer of protection that bacteria have from the environment and other types of organisms that don't have enzymes that are compatible with their proteins. This cell over here in this diagram shows our plasma membrane in green. And just external to it again is that peptidoglycan layer that we're talking about. And this cell envelope also has a capsule layer, which again is made up of sugars and proteins that is even more external typically to peptidoglycan. When we look at peptidoglycan subunits that will assemble into very long polymers, you'll see that they all have NAM and NAG cross-linked to one another, as well as an amino acid chain. These amino acids can be different depending on the species, but the peptidoglycan makeup always consists of these sugars and a variety of different amino acids that link together again to form that mesh-like network. When we look at the polymerization events that create peptidoglycan, we're having these small subunits that are going to be linked to neighboring ones by these little amino acid bridges. As they get extended and attached to one another, they're eventually going to form a very thick, highly concentrated, solid wall that is very difficult to penetrate. We talked very briefly about the Gram stain, of course, but now we're going to talk a little bit about the characteristics of cell walls specifically that allow us to characterize or differentiate cells into two groups, being Gram positive or Gram negative. The designation is based on the peptidoglycan content in these bacterial cell walls. We did talk about this briefly, but just to review, if your cells are Gram positive, they're going to have a very thick peptidoglycan layer, so 20 to 80 nanometers thick. The cells also tend to have lower complexity, so less variety in terms of protein content and other factors that are embedded in their membranes and in the cell wall itself. If you are a Gram-negative cell, you have a thin peptidoglycan layer that is usually 2 to 7 nanometers thick with a very high complexity. There is an outer membrane that's external as well. We're going to show a couple of photos here to really start to hammer these things down for you. So this is an important diagram that I hope that you will take a pretty good look at here. And these are the differences between Gram-positive cells and Gram-negative cells. Let's look at Gram-positive first. So, very important off the top, Gram-positive cells have an envelope that has two layers. We have that thick peptidoglycan layer that we discussed and a single cell membrane. And you can see that the cell membrane is internal here. On the other hand, Gram-negative cells have two plasma membranes. So it's like a little sandwich, if you want to think of it that way. You've got your external cell membrane, you've got your internal cell membrane, and in between the two here, you can see that much thinner peptidoglycan layer. So cells that are Gram-positive have that much thicker peptidoglycan and are less complex, only two layers. Gram-negative, on the other hand, are more complex. Look at all the extra things that are here in the composition of this envelope, and it has that thin peptidoglycan layer as well. This is a great diagram showing some microscopy where you can specifically see the colored peptidoglycan layer in Gram-positive cells, which is very thick. It corresponds here to this little schematic. And the plasma membrane, the single one, is internal to that. Okay, so you can distinctly see those two layers. Over here, on the other hand, we have the outer membrane, which is shown in that light yellow brownish color. We've got the peptidoglycan layer, which is this little skinny thing in between, and then internal to that is another plasma membrane. Okay, so Gram-positive over here, Gram-negative on this side, 2 layers versus 3, less complex versus more complex. We'll talk in more detail here first about Gram-positive cell walls because they are less complex. And they're going to be, again, primarily composed of peptidoglycan, that very thick layer. As a result of that, they're going to have a ton of teichoic acid present, which are in turn bound to those membrane lipids of the plasma membrane that are internal to them. There are also a large amount of what is called lipoteichoic acid molecules as part of Gram-positive cell walls that do protrude out of the peptidoglycan layer and tend to carry a negative charge. So for gram-positive cells, they overall tend to be negatively charged. You can see in this diagram here a nice schematic that shows that internal plasma membrane, those conjugations of the cell wall and its components to the plasma membrane to anchor it, and then these large molecules that span all the way out into the extracellular space. And these are the ones that we just mentioned, these purple sticks here that are negatively charged and help this cell wall to carry an overall negative charge. As you might expect for any cell, they're going to have different proteins and other things present in their cell walls, since they're the most external, that allow them to interact with their environment, whether it's for adhesion to a surface or to other cells, whether it's enzymes that are helping to build or to repair the cell wall if it becomes damaged, or even in some cases, different kinds of toxins, right? We talked about how cell walls can have pathogenic aspects to them. So some proteins that are integrated into the cell wall may in fact make gram-positive cells pathogenic in nature due to the fact that they harm the host, or the immune system in hosts will detect them and create different types of symptoms based on that detection as non-self. Gram-negative cell walls are different altogether. They are considerably more complex, with a small peptidoglycan layer sandwiched between two different cell membranes. You can see this nice little cross section here of the cell wall shows this nice little delicious looking tuna sandwich, where you can see expanded over here two different membranes outside of that small peptidoglycan layer in the middle. When we discuss gram-negative cells, one of the cardinal proteins that is most important to us is lipopolysaccharide, or LPS, and these are found in the outer membrane embedded there. We did discuss them very briefly a while back and mention that they are actually fever causing. So they are what can induce fever in a host if you do have an infection with gram-negative cells. Breaking this down a little bit further, in between these two cell membranes and where the peptidoglycan layer resides, there's quite a lot of periplasmic space, which is simply described as the area that's in between the inner membrane and the peptidoglycan layer, and then subsequently the peptidoglycan layer and the outer membrane. That is actually 20 to 40% of the cell's total volume. So you can see that there's a lot of maneuverability in here for various reasons. There is also periplasmic space in gram-positive cells, but it is not quite as expansive as what we see in gram-negative cells. Where we have teichoic acid and lipoteichoic acid in gram-positive cell walls to articulate the membrane to the wall, we have bronze lipoproteins in gram-negative cells. So similar function, where you can see them spanning the gap between the peptidoglycan layer and the outer membrane here, and that again allows to stabilize the envelope as a whole here. Lipopolysaccharide is a very large and complex molecule that has a lipid and a polysaccharide sections. The lipid component here allows it to anchor itself and embed itself into the plasma membrane because, again, if it has properties that are similar to the membrane, it can attach there very nicely. And it also has a polysaccharide side that can be different depending on the species. So this is the portion that is typically going to cause problems in the host and what will help to induce that fever state in some hosts when lipopolysaccharide is detected. So looking here at a 3D model of LPS, we have that hydrophobic component, that's the anchor that would be sticking into the membrane, and then there's this very large polysaccharide part that sticks out into the extracellular environment here. LPS also creates an overall negative charge to the cell wall, so gram-negative cells are also negatively charged. It also aids in attachment and adhesion, and especially in biofilm formation. And if you're wondering what a biofilm is, it's simply a layer that can form that is very difficult to remove from different types of surfaces. One great example of a biofilm is dental plaque. So when many bacteria get together, they start to secrete different sugars and other factors, and they essentially glue everything together very compactly so that nothing can really get in there. You can brush forever and you'll never remove plaque unless you physically get in there and scrape it away with something a little bit more abrasive. So when you go for a dental cleaning and they're scraping your teeth for 30 minutes, removing all of that dental plaque, that's a biofilm. So LPS does contribute to that formation of biofilm as well. We also have the aiding in creating permeability barriers. As you imagine, LPS is quite a bulky molecule, and things like steric hindrance still exist in the bacterial world, so things are not able to access the cell surface as easily if you have a lot of stuff in the way. And there's also how LPS contributes to pathogenicity because that O side chain can be swapped out. So, for example, if a bacterium infects a host and the host starts up an immune response against a specific component of the O side chain, and the bacterium is getting slaughtered by the immune system of the host, all it has to do is chop it off, put something else on there, and all of a sudden the immune system doesn't recognize it anymore. So it's very adaptive in terms of immunogenicity and pathogenicity in hosts. And most importantly, it can act as an endotoxin. An endotoxin is a component of a cell that functions to cause toxicity in a host, but that's attached to the cell. So it's a part of it. It's not secreted or released elsewhere, not put into the blood system, not released into the extracellular environment, but it's part of the cell itself. So LPS does serve as the most potent endotoxins that is known. Now, you might be wondering if LPS is so good at doing all of these things that keep things away from the cellular surface by being big and bulky, then how does anything ever get into a cell? And how do things actually permeate through two cell membranes and a cell wall to get inside gram-negative cells? Well, we do have porin proteins, which are also transmembrane proteins that will allow selective passage of molecules through the different membranes, through the periplasmic regions, and eventually into the cell. As you might imagine, getting things through so many different roadblocks and barriers has to be highly controlled and very well orchestrated in order for those cells to continue to survive. Looking now beyond the cell wall, still talking about the cell envelope though, is that capsule or slime layer. So this is typically external to all cell walls and cell membranes that are present, depending on the species. Not all cells will make a capsule or slime layer, but typically they're going to be called a glycocalyx. This is the sugar coating that is a layer of polysaccharides and can also have protein incorporated that surrounds the entire cell. So this is something that's produced in the cell and then gets secreted out onto the surface to coat it. There are two kinds of glycocalyx possibilities. We have capsules that are extremely organized and firmly attached to the cell wall. So you can see here, if this little yellow bit inside is our rod-shaped cell, the capsule that is external to it is going to be very firm, very compact, very highly organized. A slime layer is just essentially a bunch of loose goo that gets released by these cells and still provides a similar function, but is going to be looser or runnier and only loosely attached to the cell wall as a result. A capsule will serve several functions, primarily protecting cells from water loss, depending on what environment they're in, of course. It can block access to the cell surface, so things that are pathogenic to bacteria, for example, like different viruses, simply cannot wade through a large sugar coating. It also makes them sticky because it's made of sugar, right? So they can attach not only to different surfaces, but also to other cells. In terms of human biology and human health, the cell capsule also prevents phagocytosis. If you need a refresher on phagocytosis, it's simply the engulfment of foreign entities by specialized cells as part of the immune response. So our immune cells detect something foreign and they eat it. They say, "You're coming with me, you're my lunch," and then it gets destroyed. But with phagocytosis in capsules, the capsules can sometimes coat different features of bacteria that would allow for our immune system to recognize them. So it's almost like if they've put on a disguise when they start to secrete a capsule, which essentially protects their true identity. So it makes it more difficult for our immune system to see them and to act in turn to destroy them when it's necessary. So we'll look very briefly here at archaeal cell walls. So instead of peptidoglycan, what they have are variable and different from bacteria and something mostly made up of what's called an S layer. The S layer is going to be similar to peptidoglycan in terms of structure. It's made up of sugar and also proteins, and it's also going to be separated from the plasma membrane in a similar fashion to what we see in gram-positive and negative cells as well. Some archaea can also have even more external layers that can be made up of protein, or also something like methanochondroitin, which is a polysaccharide layer that is very similar in structure to animal connective tissue. The cell membranes in archaea are quite unique, and this is primarily because of the fatty acid chains that are found in their lipid membranes. In fatty acids for eukaryotes and prokaryotes, we only ever see straight chain fatty acids or some degree of unsaturation, as we mentioned earlier. Archaeal cell membranes will introduce even more diversity in terms of their structure. You can have fatty acid chains that are branched. So you can see in this example here, there are lots of little methyl groups sticking off of the main chain to help to create more space between lipids in these membranes. Or even in some cases, there are ring structures incorporated. So if you imagine what we discussed earlier, where introducing a single kink with saturation can help to push phospholipids further apart, when you think about where archaea live, in the coldest or the hottest places, they're going to need to be able to adapt to those environments by altering their cell membrane structure. And by making themselves extra bulky, they can even further adapt to very cold environments and prevent themselves from freezing too quickly. The cytoplasmic matrix in prokaryotic cells is pretty much the cytoplasm. It's the fluid that is internal inside of the cell envelope and will suspend the nucleoid region, which is the DNA region, and also the ribosomes, any kinds of inclusion bodies, which we did mention, which are kind of like specialized internal structures in certain types of species that have specific roles, like magnetosomes or some that are photosynthetic, as we've seen in the phylogenetic survey stuff. And like you might expect, the cytoplasmic matrix in bacteria is going to be approximately 70% water. It's an aqueous environment, which matches exactly what you'd expect in other cells like eukaryotic ones. Let's talk a little bit about ribosomes. Ribosomes are found in eukaryotes and prokaryotes, but they differ in terms of the size of their subunits. If you've forgotten, ribosomes are important in protein synthesis and are made up of two different subunits, one large and one small. So this is showing the general structure of a ribosome with the large subunit on top here and the small one on the bottom. And this, if you don't recall, is part of the translation process, where we have messenger RNA that's produced from DNA transcription that gets read by ribosomes, and then you have the conjugation of amino acids based on the codons that it detects in order to form long proteins, which are then folded into whatever they might happen to be. In the nucleoid region of bacteria, there are going to be chromosomes and proteins, and it's again really important just to remember that this is not encased inside of a membrane. There is no nucleus. The nucleoid region simply means that it's similar to a nucleus, but not quite, because it's not membrane bound. Bacteria also differ from higher organisms because you typically only have one chromosome, which contains DNA, and that can be linear or it can be circular as well. DNA in these cases can be 250 to 700 times longer than the cell. And you might be wondering, well, how does it all stay in there if it's not even encased in a membrane especially? And this is due to supercoiling. So you have a ton of DNA here that gets wound up into very, very compact units that are bunched together to form this very nice tight nucleoid region in the middle of the cell. Another feature of prokaryotes that is not seen in eukaryotic cells is the presence of a plasmid. So plasmid DNA can occur. It does not always occur in all bacterial cells, but what it is a set of external genes that are not incorporated into the regular chromosome. These are typically adaptive genes, and we see these a lot in exchange between different types of bacterial species. So for example, you can have genes on a plasmid that help with something like antibiotic resistance, or maybe it codes for proteins that help with temperature adaptation. They're typically adaptive. So certain cells that have these plasmids tend to persist longer in different kinds of environmental stresses, and plasmids can be replicated and shared with other bacteria to give them the adaptation as well. An important distinction here is that plasmid genes are non-essential, but do tend to be helpful, especially in that adaptation sense. Flagella are long hair-like appendages that extend from cells and also allow motility. These can spin, they can whip, they can do a variety of different motions, but essentially they propel bacteria in a specific direction intentionally. They're very difficult to see in light microscopy unless they are stained. And flagella come in all different types of arrangements. You can have monotrichous bacteria that have a single flagellum at a single pole. So some examples of those are right up here. We can have amphitrichous, where we have two flagella, one at either pole. So if you imagine this little cell with another little flagellum sticking out of the other end of it, that would be amphitrichous. We can have lophotrichous, which is like this fellow right here with a bunch of different flagella at a single pole. Or we can have peritrichous, where we have a ton of different flagella that come out all over the place and make for a very boisterous collection of these hair-like projections. So these are some good up-close diagrams. We're going to talk a little bit about flagellar stains and things in the lab as well, but the takeaway here is that there is diversity and variability depending on the species, but that this movement is intentional and well controlled by bacteria and gives them some autonomy over what they're doing on a given day. Sessile bacteria don't usually have flagella, but some bacteria can go back and forth between that sessile state and a motile state. So they can have a flagellum, they can shed it, they can stick it back on when they need it. That's an adaptation that some bacteria do have. Flagella tend to be kind of like a propeller, so they generally whip in a direction and allow for bacteria to move forward at all times. So you have runs and tumbles, which essentially encompass forward thrusts and also random directions as well when you have multiple flagella. So you can see in the case of monotrichous or peritrichous bacteria, they have a tendency to arrange and propel themselves in a single direction. Whereas if you have many flagella that are all kind of going independently of one another, or even in the case of Fimbriae, you can have bacteria that are just kind of tumbling around in their environments. Spirochetes, as I mentioned, can corkscrew through different liquids, and this is due to a specific protein that they have that is contractile in nature. So they have this axial filament that runs along their length, and when it is contracted, you will see the entire cell rotate and contort, which will allow them to be able to move through whatever medium they are a part of. And last but not least today, we'll talk about endospores. Endospores are specialized structures that allow cells to persist in environments that become unfavorable. So if they are free living, they're happy, they're vegetative, dividing, and all of a sudden something changes to the point where they're unable to survive, one last ditch effort for them to be able to persist is to form an endospore. The endospores themselves are very resistant to stresses. They are very rarely affected by things like heat, radiation, chemicals, even things like drying out. So they tend to help that bacterium in its dormant state come back after a long period of time when things are more favorable again. So in these diagrams here, you can see vegetative cells are just normal. They're ones that are happy, their environment is favorable for them, they are readily dividing. And then if environmental conditions start to change, they're going to start to create that endospore as a saving grace. The endospore will contain things like their DNA and some essential proteins. And it's almost like if you think about seeds that get produced by plants. And we talked about this as well, but versus creating a new individual, it simply preserves the individual that turned into the spore in the first place. So this individual cell can sprout back up again to life when the environment is restored. And you can see in these diagrams here, these cells are creating these endospores. You can see them forming. These are terminal, meaning that they're at the tips of these cells. And then in this case, this is a mature endospore where that particular cell has put all of its important stuff inside and has temporarily died, if you want to put it that way. It's gone into that dormancy state. Endospores are a big problem when we think about different types of agricultural processes or even in medical microbiology. Because they are so long-lived and so long-lasting, they can persist in soils for many, many years. So if there are different bacterial species that cause plants to become sick or infect plants that can no longer be produced or fed to livestock, for example, That's a huge issue because that soil will be unusable until those endospores are eradicated. But if they're resistant to virtually everything, they're very hard to get rid of. The other thing is medical microbiology, where we think about treatments of things, and endospore forming bacteria are very difficult to treat for the same reasons. If your medications can't penetrate them, cannot affect them in any way, you can't get rid of them. Not all bacteria form endospores, but it is important to note that the ones that do cause a lot of problems in terms of treatments. We also have a lot of different nomenclature depending on what part of the cell that the endospore form in. They have different names. So you have central, obviously, which is in the middle, subterminal, which is kind of toward one end but not quite there, and then you've got swollen sporangia and terminal. The basic structure of an endospore is exactly what you're probably thinking. All of the important stuff is in the middle. So you've got your nucleoid with your DNA, your ribosomes. And then as we head out towards the outside, you've got a core wall, the cortex, which is made up of peptidoglycan, a spore coat, and an exosporium. So tons and tons of different things between this very precious DNA and the outside world. When those environmental conditions do become ideal again, we have an activation sequence that can happen where the spore is going to prepare for germination. This is typically activated by heat or water or some other signal that will allow that endospore to know that the time has come to return to the vegetative state. And again, if you're wondering what vegetative is, it's just that metabolically active state where cells are actively dividing. Then there's the germination step where we have the dormancy state go away. The spore is going to start to swell and rupture, which is going to release again all of the important components back out into the environment. The outgrowth is the third bit where the protoplast is going to start to produce new components and will redevelop into an active cell. So over time, this one cell that's been dormant, it could be for a month, it could be for a day, it could be for many years, can return to the living state. It's a pretty neat system. And that just about does it for this lecture. We've gone over quite a lot of material here to kind of reinforce things that we already knew, to expand on things a little bit more, and hopefully you can spend some time with this and again get these basics so that as we go on in future weeks, when we start talking about topics that delve into these things or require the knowledge of these a little more, you'll have all of this stuff at the tip of your brain and ready to go. As always, if you do have any questions, you know where to find me at laurar@nipissingU.ca. And thank you so much for your attention and take care until next time. Bye for now.